Today we'll discuss on the breeding methods, pedigree selection, right? Earlier we have discussed backcross breeding. All these are breeding methods to handle segregating materials. So breeding methods used for handling segregating generations are pedigree method, bulk method, backcross method. Backcross method we have discussed, and we have also discussed the segregation ratios there, and epistatis also, digenic ratios we discussed while discussing on backcross. Backcross breeding is a simple method, whereas pedigree method is a little bit, the more cumbersome, you can say, compared to the backcross, because you have to keep the records on that. So the objective here is to any segregating material handling. It is mostly in the self-pollinated crops, and selfing is happening, and they are segregating after the F2 generation, and I mean after the F1 generation. So F2, the material starts segregating, and there we are making the selections, but the material is tending to be homozygous after every self generation. So here the ultimate objective is to develop pure line, right? If it's self-pollinated crops, pure line, or if it is a cross-pollinated crop, it is going to be an inbred line, right? If you self in cross-pollinated crop, it may not be a pure line. It can be inbred line, right? The difference between the pure line and inbred line, pure line, it is in self-pollinated crop, completely homozygous for all the genes, whereas inbred lines, it is the derivative of after the selfing, several generations, but it is in cross-pollinated crop. It may not be 100%. All the genes may not be homozygous conditions. So the pedigree selection is one of the important and basic method of breeding to handle segregating material. Pedigree may be defined as a description of ancestors of an individual, and it generally goes back to some distant ancestor or ancestors in the past. Any number of generations go back. Pedigree would describe the parents, grandparents, great-grandparents, and so on. So the relation you can bring out, how the present material, whatever line is there, it is derived from which parent, which are all the parents involved in previous generations. Like that, you can bring the relationship or trace back its ancestor. It is useful by finding out two individuals are related by descent. Any individuals, if you take into consideration whether they are related to one another or not, you can answer this question by following pedigree method. And if they are related, yes, they have a common parent, and their ancestors also, many common ancestors are there, and they have same genes in common. They are having same genes in common. That's a implication or meaning of it. In the pedigree method, individual plants are selected from F2 and subsequent generations, and their progenies are tested. A record of all the parents' offspring relationship is kept, and that record is known as pedigree record. How the pedigree record is maintained? Just give you one example here. For example, today I have PMR12. This is a live example, I tell you, working at IHR. This is a PMR12, the line. How this PMR12 is derived. You look at this you have to, after consolidating all the information of all the generations, you are going to write the pedigree of this particular line which you have today, right? So how this is derived, you can, by going through this record, pedigree record, you can understand. For example, how to read it or how to write it also, you can get the idea. So to start with how this PMR is developed, to start with, 11th line is crossed with 8th. What is the 11th? 11th is a Panth C1 chili variety, and this is crossed with 8th. It means 517B. It's the action of capsicum beccatum variety pendulum wild species used to cross. So they got a hybrid. Hybrid was back-crossed for BC1, that is Panth C1. So this 11th line was crossed. So that is the This is the first generation F1. Then F1 is backcrossed with the Panth C1 chili, that is BC1. And then you get a material, right? You get a seeds from this BC1, you raise it in the next generation. You got you will sow the seeds in several rows. So the next generation, it is the 10th row, right? So the 10th row, not specifically mentioned about the plant and all, it was crossed with 483-4. So 483-4 is capsicum varieties. It's not the same parent they used in the next backcross generation. It is the capsicum parent was used for the as a recurrent parent, right? 483 plus. This is just crossed with the line derived after this 11 into 8 BC1. Okay, so from this, okay, you backcross and you get a seeds. So in the next generation, you get several seeds. You sow them in rows. It is the second row, 8th plant was selected, and the second row, 8th plant was selected. Seeds were collected, and it was forwarded in the next generation, where several lines, combined, and those are around the several lines, 241 line, right? 241 line. It was put in 3 rows, right? Third row, 7th plant. So the seeds were collected from that, and it was forwarded in the next generation. So next generation, once they raise, it is then the 9th line, which is shown in several rows. Third row, 3rd plant. That plant was that was crossed with 934. It's one more capsicum line was used here. 934C, whenever it is written, capsicum pollen was used, crossed with this, and they got a seeds. Those seeds are raised in the next generation, and several lines they are raised simultaneously. 171st line, which is shown in many rows, and the 3rd row, 2nd plant, seeds were collected, and those seeds were raised for the next generation, where next generation, 192nd line, 2nd row, 7th plant was used, and it was crossed with selection 16C. Is one more capsicum line, right? Capsicum line was, the pollen was used and crossed. Seeds they got. The seeds were sown in the next generation, where the 16th line, right, 3rd row, 6th plant, the seeds were collected. It was pushed to the next generation, where 214th line, 4th row, 1st plant. It was crossed with selection 13C. Capsicum line was used, pollen was used, and they got a seeds. Seeds were, the further raised in the next generation. 55th line, 2nd row, 8th plant, right? And this plant seeds were collected, and as it is, the seeds were carried forward, and the next generation they raised this lines, and the 61st line, 1st row, 6th plant. This seed is nothing but your PMR term. Okay, this is the meaning and how you have to write also, right? So here some description was given. First year, second year, what is that? First year or first season? It's a first season, second season, and then this is the 3rd season. This is the 4th. This is the 5th, and this is 6th, and the 7th, 8th, 9th, 10th, 11th, and the 12th, right? So 12th seasons, you can say generation also. So this is what PMR they got into. This is how you have to write. And this is a bit complex, right? Backcross was involved, modified backcross was involved in segregated material. Record was kept, and that is a pedigree record. Now, if you look at the simple pedigree record, for example, I got 9214 line is there. Now, how this has derived? Of course, this 9214 means it is a year 1992 on the 14th cross number. Okay, that they got it, and how this was forwarded. From that the F1, they got the crosses there 1 into that's 14th is the cross number, and the seeds were collected and selfed in the next generation. They put it, that is F2, right? This is F1 and this is F2. 4th row, 5th plant, F2. Seeds were collected, self seeds were collected, raised for the next generation, that is F3. 6th row, 7th plant. Next generation, F4, fourth row, fourth plant. The next seeds, some seeds collected and put for the next generation, that is F5, third row, sixth plant. Next F6, second row, sixth plant. Okay, this is how you can write and identify. I mean, understand how this pedigree record is made. This is simple, simple pedigree breeding method followed here. Previously, that was backcross, modified backcross. So in pedigree selection, only important characters should be recorded because it is a cumbersome. You cannot have so many characters. You need a lot of resources, human involvement will be there. Only promising, it's one is the characters. The characters of the plant which are recorded, you are recording, it should be kept minimum. And then second aspect is only promising progeny should be included in the record. That means not everything you can keep in the record because it requires a lot of time. Then the third point is pedigree record must be accurate, not that anything. That's why it is said you reduce the number of characters, you reduce the number of progeny so that you can maintain the accurate pedigree record. So pedigree record must be accurate. So it's a key role is that keeping no record is better than keeping an incorrect record. Something you are going to misguide. If it is recorded, you keep it accurately. Then only it will help you. Otherwise, simply misguide. That's why keeping no record is better than keeping an inaccurate record. Where this pedigree method can be applied. Most commonly used in for the selection from the segregating generations from the cross in a self-pollinated crops, commonly it is used. And it can be used to correct the specific weakness of an established variety that you can use one of the parent and go for a pedigree selection. And to select the new superior recombinant types, to select the transgressive segregants, that means you can expect the parents falling outside the range of the progeny is falling outside of the range of parents. You will get the situation. Such plants are they are superior to the parents which are used, right? That's why it is also referred as the transgressive breeding. That means transgressive segregants you will get better one what earlier it was there. And you can employ this method to improve the specific characteristics, the disease resistance, maturity you want to alter or get earliness. You want to have dwarf plants or anything you can use the pedigree method. So general procedure, first year, two parents, P1 and P2, they are homozygous. P1 is also homozygous and P2 also homozygous. Maybe they are differing with genes. One P1 can be homozygous for one gene, homozygous dominant for one gene, and P2 is homozygous dominant for some other genes. Like that, the advantage of this dominant genes, you can get it here. So two homozygous parents are crossed. So selected the parent planted in a crossing block, and the crosses are made. You make the crosses, get a seeds, use it for the next season. Second season, the seeds, whatever you got in previous season, they are nothing but F1 seeds, and 10 to 30 seeds space planted from each cross and harvested in bulk. Right? It's only number of plants you want to have. That's important. Otherwise, there is no difference in the plants of F1, right? Because these are homozygous. Whatever any kind of combination, zygote formation, it is going to be heterozygous for any differing genes in these parents. So that means it is a homogeneous material. Heterozygous, each individual is heterozygous. Whereas F1, if you look at so many plants are there, they're all genetically alike. Okay, so that's why it's only multiplication, number of seeds you need for the F2. Based on that, you can have. You need not have too many plants there. Okay, then the third season, that is whatever the seeds you have got, bulk seeds you have got from F1, you are raising in the third season, that is F2, right? Here, 2,000 to 10,000 seeds are space planted. It again depends on the annuals, like the cereals and pulses. Their spacing is very close to the recommend. But vegetables, when you consider the number of the spacing is more and it requires more space, you can reduce the number. So if you plant 2,000 to 10,000 seeds, you can select 100 to 500 superior plants out of this. Okay, are selected. and their seeds are harvested separately. Now, whatever single plant you are getting, I mean selecting, the seeds of that plant you have to collect separately, right? So then, okay, F2 onwards pedigree record should be done carefully. Now plants are differing, plants are segregating. You have to maintain the record that which plant you have got. I've explained you earlier that pedigree record of maintaining which row, which plant, which line, all that you have to maintain. Then the selection ratio, always you keep it in mind. 1 out of 10 or 1 out of 100 F2 plants, right? So one plant is selected, that is going to be pushed for the next generation, that is F3 generation. So if there are 100 F2 plants, you select only one. So, or you can also have a range, right? Like a 10 plant or to 10 plants one cell you select. It depends on the, that depends on how distant the parents are. If the parents are distant, you can have more varied plants you can get it. So you can select 1 out of 10 rather than 100 out of. And again, it depends on your crop spacing, how much you can handle, all that you keep in mind and decide this number. Objective is use the heritable and morphological characters. Very important. What visually you could see, use those characters as a basis for selection. And also, they should be heritable, meaning they are controlled by fewer genes. Everything is heritable, that is different. Environment interference will be there. But the thing is, characters which are controlled by fewer genes, we refer them as heritable, right? General. Fewer genes means less of environment interference. What you see, you can believe that it is because of the genes. Okay. Genetics, I mean the heritability of the character is very important. So use the knowledge of genetics in selection. This is very important. So then only will be efficient plant, but otherwise you're simply wasting the resources. Do not select too many plants from F2 generations. So handling them is going to be an issue, right? Then fourth generation, fourth season, and the third generation F3. Now whatever the plants are selected there, 1 to 500 or 1 to 100 number of plants are selected and collected seeds separately. That each separately collected seeds should be planted in a row, separate row. Individual plant progenies are planted. That means one plant seeds, whatever there, one row it should be, right? It's a row. The line, this is one, actually it is representing the row, one row. From this, the superior plants are selected. Each progeny should have 30 or more plants. One row means around 30. It depends on the crop, I told you again. So outstanding plant from inferior progeny also can be. Now the entire row you are referring it as a progeny. Like so many progenies are 50, 100 progenies are there. Out of this, which is the best, you look at all the plants, progeny and you make. Otherwise, if there is something within a row, something is very outstanding compared to other plants, still it is segregating, right? So in that case, as that particular plant you can separate, select separately. Number of superior progeny small, the whole cross may be rejected, right? If all these progenies are not doing well, then entire class is rejected. Don't go further. Don't go for F4, F5, right? You can take a decision. So remember, number of F3 plants selected should be less than the F3 progenies. So next, whatever the plants you have selected here, you are collecting the seeds separately and you are raising it in the next season, that is fifth season, and that is referred as a fourth generation. Again, here also row. One plant, whatever you have collected seeds, it should be raised in one row. So same thing, whatever you have followed in F3 generation, same thing, all the guidelines you can follow here. If two or more progeny superior and comparable coming from the same progeny, select only one. No redundance or no duplicates there. And then the number of F4 plants selected should be less than the number of F4 progenies. You have to keep reducing. So maybe 500 is started in F2, you have to come down, right? So the F3, 100, then here like 50, you keep reducing that number of plants selected. Then the whatever the seeds you are collecting, it is going to rise in the next season, that is the sixth season, and they are referring those seeds as F5, right? Individual plant progeny planted in multi-row. That means one progeny, whatever you have selected, that is raised in 3 rows, 4 rows, multiple rows you have to raise because they are increasingly become similar. Superior plant selection from the superior progeny. So you got right. Okay, this is superior. The entire progeny is superior. All the 3 rows, 4 rows, it's good. No, in that the superior plant you have to one plant. Now, many families may have become homozygous and may be harvested. If you feel that they are getting into homozygous status, F5 you have got into, you can start harvesting in bulk because they are becoming similar, genetically becoming similar. If segregating any row is there or any progeny is there, then individual plant has to be selected. Again, here also reduce the number. The number now you should achieve to 25 to 100 progenies, depending. So keep reducing. So whatever the seeds you are collecting, you are raising in the next generation. That is the sixth generation, and it is 7th season. Individual plant progenies are planted in multi-row. What I have done in the previous season, same thing. Superior plants selected from superior progenies. Many families may have become homozygous, may be harvested in bulk. If segregation, then individual plants selected. Size reduced to 25 to 100 progenies only, right? So here you can initiate preliminary yield trial. So far, you are not considering yield as a criteria for selection. Now onwards, you have to go for that. It is less heritable because controlled by more genes and more environmental interference is there. In that case, you have to you are not going considering that. Now from this season or this generation, F6 generation, you can go for that yield trial, preliminary yield trial may be conducted. Progeny is harvested in bulk now. They have attained the homozygosity. Progeny showing segregation may be eliminated. If anything is showing segregation, it's rejected unless it is very outstanding. And such progeny individual plants may be selected, outstanding plant. Collected seeds are raised in the next season, that is 8th season, and you are referring it as a F7 generation. And here, yes, preliminary yield trial with chuck. Now the commercial chuck you have to grow along with your material. And it is now blocks, right? You are having the block. Quality tests for additional basis of selection. Now, so far, the yield, right? It's not only yield is sufficient. Now you add some more characters for cutting down, right? It's reducing. Okay, yield all are okay. All are good yielding only. But now, based on the quality, you can reduce the number. So 2 to 5 lines selected, which are superior to standard check. They should be. Then you are aiming at releasing the best varieties. 9th to 13th season, that is F8th to F12th, you have to conduct the replicated coordinated trials or multi-location trials, right? You have to conduct different seasons, different locations. You can do it, and stability of those lines with respect to yield you can study. During that time, you can also prepare the descriptors. That means when you want to release the variety, you need the characterization of that variety. How tall this, how many branches it gives, what kind of leaf it is, what is the internodal length, what is the color of the fruit, what is the the pigmentation on the stem or anything, any of those characterization, descript. They have to prepare the descriptor of that variety. And you can also go for disease and quality tests there. So we are now confirming, yes, it's very good. Now it is falling in the channel that you can push it for release. So that how the release procedure and all, you have to get the information from the zonal committees or the variety release committees. Based on that, you have to prepare yourself the entire material for release. So the 4th season, you can go for the seed multiplication and distribution of the seeds. So basis of selection, quick visual evaluation of the plants, we should go for. Morphological traits like height, leaf size, shape, head type, fruit color, days to flowering, days to maturity, and also disease resistance you can use. Example, Arca megali of tomato, which is derived, it's a pedigree selection, F8, derived from Arca vicas into IHR 554. These are the parents crossed, and F8 generation, this Arca megali was crossed. So early generation testing also suggested by Jenkins in maize. And what is that actually? the yield selection, you are going for the F6 generation or F5 or F6 generation. Now, early generation testing means F2, F3 itself, you have to go for the yield trials. So if it is efficient, it's whatever you make the selection in F3, F4, same kind of materials are representing in F5, F6. It is studies you have to do it. Like, one side F2, F3 generation selection, on another side you carry the material to F6. Same by following both the methods, you are ending up the same kind of varieties, same genotypes. And yes, you can follow this early generation testing. That saves a lot of your resources. The merits of pedigree method provides maximum opportunity for a breeder to use his skill and judgment in selection. Early generation testing can be followed. Yes, that's the efficiency. But yes, it should work. There should be correlation between these, early generation testing and the later performance of those lines in the later generations. It is suited for characters easily identified and are simply inherited. Very important. Transgressive segregants can be recovered in addition to the specific improvement. It takes less time than the bulk method. Bulk method, you are leaving it for the nature to take this, apply the selection pressure. Like some 5-6 years, simply you grow there, because of the any kind of stress like temperature increase or some particular disease or the rainfall variation. That is the selection pressure, and genotypes which are able to do well the prevailing condition only will survive. Remaining will produce. They will not be growing better, will not be producing more fruits, more seeds. So the representation will be less, and automatically natural selection is taking place. But that takes more time. So pedigree method, if you compare it to the bulk method, yes, it takes less time. It is often possible to obtain information on inheritance of the characters from pedigree code. Definitely. If you see the parents and the F1, you will get some idea which is dominant character if you look at any character. In segregating generations, if you look at what kind of, the proportion you are getting, you can judge approximately, not precisely, like whether it is controlled by one or few genes or many genes are involved. You can predict. Of course, to confirm that, you need to have, the perfect study, right? Like F2 study, you have to do it with a ratio and go for chi-square test. But yes, preliminary, some idea. Without doing that, you can get. Plants with visible defects and weaknesses are eliminated at an early stage of a breeding program. That's advantageous. And what are the demerits of a pedigree method? Maintenance of accurate pedigree record takes up valuable time of the breeder. Sometimes it may be limiting factor in large breeding programs. Breeder has so many other programs also, not only just this program, what you are intended. So in that case, it becomes a limiting factor. Selection here is laborious and time consuming. So it is difficult to handle many crosses with this cumbersome, the kind of activity involved. Success depends on skill of the breeder, how good he is, how good he has the knowledge of genetics of different characters of that crop, and how good he is in principles of plant breeding and genetics. Then only he will be, successful. So success depends on the skill of the breeder. So no opportunity for natural selection. Whatever you do it, that is going to be retained or selected. Selection for yield in F2, that is the early generation. If care is not taken to retain sufficient number of progenies, valuable genotypes may be lost in the early segregating generation itself. You have lost those valuable genotypes early itself. That's why you're allowed to go for 4, 5, 6 generation. Then only you go for the selection of a yield, like that. The achievements: Pusa early dwarf tomato variety. Early variety have been developed by crossing Miruthi into Red Cloud, and then selection made in segregating generations. They developed this early Pusa early dwarf. Soybean pedigree lines have been developed at Asian Vegetable Center, Center Taiwan. Like that, plenty of examples you can get for the varieties developed through pedigree selections. With this, we'll close it. And